you, Vania, for the nice introduction and for presenting our institute. So I want to start by welcoming and thank a lot the invitee today. So Tiago Costa, and to introduce him, he was a former student from INESC. So he conducted his PhD under the supervision of Professor Moisés Piedade from INESC and Professor Paulo Freitas from INESC MN in the area of CMOS, integrated circuits for magnetoresistive sensors interface for biomolecular recognition and neuronal recording. In 2015, he started a postdoc position in Columbia University, New York, where he worked on ultrasonic microchip technology for biological interfaces. Now, he is an assistant professor at TU Delft, and is focusing his work in new devices for minimally invasive interfaces to the nervous system. So I'll give the floor to Tiago Costa, and welcome.
Thank you, Verónica, for your nice introduction. So let me share the screen. Hopefully you all can see the presentation mode of PowerPoint. Okay. So thank you everyone for joining. So as Verónica mentioned, my research is on minimally invasive devices to interface with the nervous system, and today in particular, or as a summary of my research during my postdoc and also now at the beginning of my career as a PI, I'm focusing on ultrasonic microchips for modulating neuronal activity. So just a bit about myself. Verónica already introduced me, so this will be quick. I was born in Torres Vedras in 1985. I did, which is also very famous for its carnival. I did my studies in IST in Lisbon. Then for a brief time, I was an engineer at Toyota in Belgium. But I quickly gave up of my career in industry to come back to university, where I did my PhD under the supervision of Professor Moisés Piedade and Professor Paulo Freitas at INESC. And after I went to Columbia University to do a postdoc in the lab of Professor Ken Shepard. And since October 2019, I'm at TU Delft as an assistant professor. Talking a bit about TU Delft before I start my presentation, or the research part of my presentation. So TU Delft is the oldest and largest technical university in the Netherlands. And it has approximately 25,000 students. And it is also very well linked with the two other technical universities in the Netherlands, Twente University and Eindhoven University of Technology. So Delft is famous by having many canals. So you can see it as a little Amsterdam. It is also the place, or TU Delft, the place where some famous researchers were affiliated with, such as Antonie van Leeuwenhoek, known as the father of microbiology. Delft is also famous for Delft Blauw, which is this type of pottery, often featuring windmills and canals, like you see here. And finally, Johannes Vermeer, one of the Dutch masters, was also born in Delft. And this is one of his famous, most famous paintings, maybe, The Girl with the Pearl Earring, which is in a museum just next to Delft in The Hague. So the university, here we have a picture of a sunny day in the university. What you see here is actually the building where I work, the Electrical Engineering, Mathematics and Computer Science faculty building. So the university organized in several different faculties, as you see here. And the one where I am affiliated with is the EEMCS faculty. And in this faculty, I'm part of the Microelectronics department, which is organized in different sections, from circuits and systems, electronics, electronic components, terahertz, so all of these different types of activities. And within these sections, we do our research in three main themes: health and well-being, XG sensing and communications, and autonomous sensor systems. And then to support all these activities, we have the ELSA Coil Lab, a clean room where I'm also doing my part of my work. So I'm affiliated with specific the bioelectronics section and with the health and well-being team. So now talking about ultrasound neuromodulation. So first I'll give a brief introduction to neuromodulation and the emergence of bioelectronic medicine. After I will talk about why ultrasound, focused ultrasound in particular, is a good prospect for a new generation of bioelectronic medicine. I'll address the technology challenges and also describe some applications in neuroscience and neurology. So first, when someone has a disease, the typical procedure is to go to the doctor and to get medication, in the basically pills. So this type of pharmaceutical drugs, pretty much everybody can take it. So there's high eligibility because it's non-invasive. You just take a pill. However, it is imprecise. So it interacts with the whole body, not just the focus of the disease, and because it flows through the circulatory system. So normally, when we look at the leaflet of pharmaceutical drugs, we often see very long lists of side effects, even though here is just a short list. This is the most typical drug for epilepsy, carbamazepine. And you see here the types of side effects that you can have, such as depression, nausea, vomiting, suicidal thoughts. A similar example for Parkinson's disease. This is, again, the most typical medication. And actually here, the list of side effects goes to 3 pages long. I'm just giving you here a small list. Of course, this is not always the case. So if we have a headache, we take an aspirin, nothing really bad happens. But when talking about neurological diseases, nearly 1 in 6 people in the world suffer from these diseases. And it's responsible for approximately 12% of all deaths. And of course, this is largely dominated by stroke, which is a neurovascular disease, but Parkinson's, epilepsy, depression, and many others also contribute to this high number. So to try to fight these numbers, the field of bioelectronic medicine has emerged in the last, let's say, 2 to 3 decades. So opposing to pharmaceutical drugs, bioelectronic medicine tries to harness the electrical conductivity of the body to treat disease. So the idea is to deliver precise stimulus only at the target of the disease to mitigate side effects. So this is one of the advantages. So to try to get rid of all side effects that we normally have from pharmaceutical drugs. However, as you'll see, there is low eligibility. So because these bioelectronic medicine devices work by means of electrical stimulation, it requires surgical implantation of a stimulator very close to the target neurons or nerves. So what you see here is the typical path from discovering the focus of the disease down to a device. So firstly, molecular biologists identify targets of disease. Then neuroscientists identify the neural pathways to manipulate the target. And finally, engineers and computer scientists design a device to modulate the appropriate pathway. However, the transition from neuroscience to a clinically approved device, there are still a lot of things that need to be done there. So if you look at the field of neuroscience, where the signal pathway is identified for a specific disease, so normally we want to look at different scales of information. We want to look at one neuron, for instance, using patch clamping, which is the standard technique to record signals from single neurons, even from inside the cell, for instance. We can look at networks of neurons, where, for instance, we have optogenetics and calcium imaging to do it with light. But this requires genetic modification of neurons to be sensitive to light and to emit light when active. And then we can also do this electrically. This is a picture of a famous Utah array that is the standard for neuroscientists to record and stimulate electrically the brain. And there is already commercially available electronic microchips to interface with these high density arrays. And then this is the type of signals you get. So basically spike trains or action potentials being recorded. Then we can also look at the whole brain. So we have for stimulation, for instance, transcranial magnetic stimulation that has a high coverage of the brain or can go penetrate deep in the brain. We also have functional MRI where you can have whole brain images where you can see where activation is happening. We also have EEG, for instance. So there is these different scales that help us to look at the brain. However, So this is a good thing, right? I think that the field of neuroscience has a lot of tools from one neuron to the whole brain. And even though there are still things to be done, I think there's good tools already. However, when you look at treatments, so to go from the signal to the device, it's not, I think so, let's say, a good scenario. So this biotonic medicine field didn't start recently. It actually started in the late 50s. So the pacemaker, the first pacemaker is an example of this. It appeared in the late 1950s. Even though it's not for the nerve, it's not for the let's say the nervous system. It's for the heart, where the cells are also excitable electrically. Then the spinal cord stimulator also emerged in the late 60s, and then in the late 70s, the cochlear implant for the hearing impaired also was firstly developed. And more recently, we have, in the beginning of the century, the deep brain stimulator for Parkinson's disease, and also now used for epilepsy, and the vagus nerve stimulator that stimulates not in the brain, but in the peripheral nerve, so the vagus nerve, for epilepsy and depression. So what you see here, it's something very common things besides of the color. So they are all very bulky. So this fits in the palm of your hand. And the main reason for this is battery, because these devices, they have to stay inside the body for years. The battery has to be large, so that it can be recharged easily and have long battery life. However, one of the biggest drawbacks is that it requires surgery, and having a large and bulky device implanted into the body often leads to low eligibility from patients. So not all patients are eligible for this normally very expensive and also risky procedure. There are also non-invasive electrical stimulators for the vagus nerve, so transcutaneous vagus nerve stimulation. But since they are far away from the nerves, they have poor spatial resolution, so they are less effective. So on one hand, we have pharmaceutical drugs, which lead to lots of side effects, and their effect wears off over time. And on the other side, we have bulky, expensive, and invasive electrical stimulators that have low eligibility because of the risky surgical procedures, and so are a last resort therapy. So only when the pharmaceutical drugs stop being effective, then patients can be eligible for these procedures. So I would say that this is not a very good scenario for treatments. So many patients are left without a solution. So what can we do? So this is what we have today. This is a time course of treatment of a disease. So we start with pharmaceutical drugs, they stop working, and then patients have biotonic medicine implanted. And to give you an idea of the low percentage of patients that can have these implantable devices, for instance, in Parkinson's disease, only 15% of patients are eligible to withstand the surgery. So at this point, when the pharmaceutical drugs stop working, 85% of patients of Parkinson's cannot continue with the treatment. So I think that the way forward is to develop either wearable and precise devices so that we can have precision without surgery, or miniaturize and make the devices so biocompatible that we can inject them or insert them without surgery as well. So the idea is that we can then, from the beginning of a disease, because there's no surgery, it's easy to start a procedure already with biotonic medicine. But the dot on the horizon is, of course, with this we have reduced side effects from pharmaceuticals because we have less dosage and there's no surgery, so there's eligibility. But for me, the dot on the horizon is to get rid of pharmaceuticals altogether and start from the beginning with the biotonic medicine. So this is the motivation for my research. So this is the ultimate goal. So no side effects, no surgery for everybody that needs it. So now I'm going to talk about why ultrasound is a good prospect for this minimally invasive or surgery-free and precise therapy. So if we look at the field of neuromodulation, if we think about non-invasive approaches, we basically have two, at least traditional: transcranial magnetic stimulation and transcranial current stimulation. And on this plot, you have a comparison between depth of penetration and spatial resolution. So transcranial magnetic stimulation can go several centimeters deep into the brain, for instance, at a resolution of a few centimeters. And transcranial current stimulation can go deeper, but with even worse spatial resolution. And let's not forget that implantable electrodes, so microelectrode arrays, since they are implanted, they can go anywhere in the brain, and they can be made very small, matching almost the size of neurons. So we are still very far away from what implantable devices can do. On the other hand, if you look at imaging, different modes of imaging the body, we have here different techniques that cover pretty much the entire spectrum of depth and spatial resolution, starting from on one hand with MRI, which is already very widely used in hospitals. We have ultrasound diagnostic imaging, we have OCT, we have computed optical coherent tomography, we have confocal microscopy. So this can cover the entire spectrum. So the interesting thing is that about 10 years ago, ultrasound has also been started to be used for neuromodulation, specifically by means of low intensity focused ultrasound. And this is, let's say, a promise for this field, because now we can cover this area that was until now not filled of penetration depth and also good spatial resolution from a non-invasive, in a non-invasive way. So this is why ultrasound is a promising candidate. And what is ultrasound anyway? So you have here basically an animation of an ultrasound wave, which is pretty much a pressure wave with a frequency above the audible range, which is 20 kilohertz. And you see pretty much if you look at just on one particle, you see it's moving back and forth for positive and negative pressures, but you have then a wave propagating in space. And to be used in neuromodulation, it has to be focused, normally by a lens. And you see here a typical beam profile of the focused ultrasound. And these are the types of waves in the time domain at the focal spot that we need to produce to achieve neuromodulation. So we have to have focal pressures, normally in the megapascal range. The pulse duration of each burst is to be hundreds of microseconds, several milliseconds. This repetition frequency, the pulse repetition frequency between bursts in the order of several kilohertz, and also the sonication duration needs to last several seconds. And all of these parameters, also the frequency, as you'll see, the higher the frequency, the better the spatial resolution. And all of these parameters exceed the typical specifications of ultrasound when used in imaging. So this is like a new challenge for ultrasound device research. So, but how does it work? How does ultrasound affect neuronal activity? Well, nobody knows. And that's a very interesting research question right now. There are hypotheses, and this is the latest one by a group at Caltech, where they have found that specific mechanosensitive ion channel proteins in the cell, in the neuron cell membrane, respond to ultrasound. So first, because they are mechanosensitive, they respond to mechanical inputs, which is the case of ultrasound. And these researchers have identified a series of mechanisms that are triggered by these specific ion channel proteins. However, these conclusions are still very indirect, so it's still only an hypothesis. On the other hand, we have the effect of radiation force on the cell membrane alone. So you can imagine a membrane with a pressure wave makes the membrane vibrate, and this leads to modulation of currents between the intracellular space and the extracellular space that can lead to an activation of a neuron. Then we also have cavitation of a cell membrane, which can lead to pores forming in the membrane and even to the membrane being disrupted. And cavitation is not, it's a destructive process. So we hope that this is not a mechanism, but it's still not clear how it works. However, we know it works, because there are several papers illustrating ultrasound leading to action potentials, and you have a few examples here. So here I have a brain of a rat. We have an ultrasound beam in this direction. We have an electrode near the focal spot. And what you see here is that after the ultrasound stimulus is applied, the activity increases. So you clearly see the correlation between these two. There are several examples of this. This is another paper where the focal spot was created in a region of the brain, and this then was moved to different locations, and fMRI was used to record the response. So you see here, this was due to this focal spot being moved in different spots. So you see activation, but not only in the brain, also in peripheral nerves. This is a group that I collaborated with at Columbia University, focusing ultrasound in a nerve in the leg of a mouse, and then recording EMG responses with the electrodes nearby. And what you see is as the focal pressures increase of ultrasound, the success of activation of EMG activity starts to increase, and for certain conditions is 100%. So these are all verifiable results, even though the mechanisms are not perfectly understood. So what are the technology challenges here? So what we need to do to bring this to patients? So this is a challenge in my view, and maybe you can already see. So these ultrasound transducers typically have this form factor. So you require basically a computer and a handheld focus transducer, which is normally a piezoelectric disc with a lens, so that you can focus, you can drive it with the signal at the resonance frequency of this piezoelectric transducer, and the lens focus the wave in a specific location. One of the great drawbacks is not only the size, but to move the focal spot location to precisely target specific neurons, this has to be moved really normally, not by hand, but normally using precise micro manipulators. So we cannot imagine this being used in a patient, right? Because with movement, even we have a position correct at the beginning, with movement or the pulsation of the brain, focal spot will no longer be in the right location. So the challenge is twofold. So first, miniaturize these gigantic boxes into microchips, so that we can either make it a wearable patch or insert it into the body in a minimally invasive way, and try to increase the spatial resolution also from millimeter size to the hundreds of micrometer or below, to continuously push spatial resolution towards the size of single neurons. And the advantage that I didn't mention is the ability to change the focal spot location electrically or electronically without relying on movement. And this is achieved by beamforming, as I'm going to explain. And of course, what we want to target is two different regions of this, let's say, diagram. First, for patients, we need to provide certain depth of penetration. So we want to operate in this region, in the several centimeters of depth, to target, for instance, deep in the brain. But we also want to understand how this neuromodulation in ultrasound works. So we also need to develop technology to study really the fundamental mechanisms of ultrasound neuromodulation, so neuroscience application. So ultrasound waves can be focused electronically by using phased arrays. And phased arrays are a very old technique that was used in radars, and also then adopted by the ultrasound community to make ultrasound imagers. And this is basically, instead of having a single disc of piezoelectric material, we divide it into small elements, and then we drive them with electrical signals that resemble the lens, that have the same kind of curvature of the lens. And you see here that kind of focusing profile in the electrical domain, and what we get in the acoustic domain or in space is the focal spot being created in a specific location in space that matches pretty much the geometrical focal spot of this lens. And the nice thing about this is that, okay, the electronic signals, we can change them. So we can pretty much make a lens that has any shape to place the focal spot anywhere in space within a given volume. And this is how it's implemented. So a very, let's say, high-level description. So we have these microchips that I have a CMOS chip with all the electronics to generate these specific phases. So at the input, we have, for instance, a clock signal, and then we apply a specific phase to make a lens. And this happens for every single channel in the CMOS chip. And then on top, we fabricate an ultrasound transducer to convert the electrical signal into the acoustic domain. Then what we want is to achieve something like this. So we create these lenses in the electronic domain and we have the waves propagating in space with the same time delays until we have a focal spot. And there are here a few important parameters about these waves to discuss. First is the diameter of this focal spot, which gives us the spatial resolution of the stimulation. And it basically increases, sorry, we want to decrease this number, so we need to increase the area or the aperture of the phased array, of this 2D array. We want to increase the frequency of ultrasound to decrease the wavelength. And also, the deeper we go, this focal spot diameter increases. So there's a certain limit in the depth for the specific resolution. So the maximum focal depth, which is also important to define. So how deep can we go? And this is also, it also improves with larger apertures and with larger frequencies because lambda decreases. And the same happens for the focal gain. So the gain from the surface of each piezoelectric transducer to the focal spot. And again, larger apertures, larger frequencies help us. So the challenge here is on how to increase the aperture. So how to increase A to improve all of these parameters. But a larger A means we have more elements in our array. So it means we need more electronics, which requires more power. And you can see where this is going. So it's becoming more difficult because of power dissipation. And if we increase frequency to decrease lambda, you see here that each channel needs to be spaced by lambda over 2. This is to avoid grating lobes, which are side effects. You can see it as unwanted focal spots appearing in different locations in space. And to avoid these grating lobes, we have to space each element by lambda over 2. So if we increase frequency, the spacing decreases, and it becomes very challenging to design the electronics and to fabricate ultrasound transducers in such small spaces. So these are basically the trade-offs between the design of the devices and the performance. And this is a block diagram of how, in a particular case, one implementation that I did in my postdoc. So we have a clock at the input. First, there's a coarse phase control that generates 7 different clocks. I'm going there after. So 7 different clocks that then are broadcasted. They are routed to all of these channels in the 2D array. And in each element of the array, there's a fine phase control. So it refines the phase. And then a driver to boost the driving voltage to drive the ultrasound transducer. And there's here an example of the clocks that are used. So from 0 to 180 degrees in steps of 30 are then sent to all channels. In each channel, there's a fine adjustment with a resolution of 7.5 degrees. And then the voltage is boosted normally to 20 or more volts, ideally, to achieve more pressure at the output. So you have here a few specifications that normally we want. Driving voltages up to 30 volts. The peak currents are very high, but this device is not continuously operating. So this will be then averaged to a value below 100 milliamps or even below 10 milliamps. And the timing resolution that is needed is in the order of nanoseconds to have precise focusing. So this is the version 0, let's say. This was done during my postdoc. So we have here a CMOS chip, which is an implementation of what I just explained for a 26 by 26.
Sorry to interrupt. I don't know if
Yes.
it is only a problem of my connection or is everybody listening well to Tiago?
Yes, Verónica.
It's okay. Once in a while it goes out.
No
but
problem, yeah.
yeah.
Moving well.
Okay. All right. Maybe it's just okay then. All right. As I was saying, this is the CMOS chip that I implemented, a 26 by 26 array of driving channels. And that was tested. So this is, you see just a few results. For one specific channel, swept all the phases from 0 to 360 degrees. So you have here from 0 to 180, 180 to 360. And here you have the transfer function from digital input to delay. Here in nanoseconds, a frequency of 10 megahertz was used here. And then I programmed the chip just to see how it would work in simulation. I programmed the chip to output signals that would correspond to a focal spot 2 millimeters deep in tissue. So and I recorded all of the signals in the probe station all of these outputs. And you see here the examples for row number 13, all the columns, where you already see the focusing of all these driving signals. And then I did simulation in MATLAB, first with ideal signals, and then with signals measured from the chip, and I got the same profile. So it means that the electrical part of the chip was working fine. And then, of course, what about the transducers, right? This needs transducers to transform these electrical signals into acoustic waves. And I've been talking about piezoelectric transducers, but are in fact two types of transducers that are most used for these applications, imaging and neuromodulation. Of course, based on the piezoelectric effect, most commonly PZT, lead zirconide titanate. And we also have MEMS-based transducers, in this case, capacitive micro-machine ultrasound transducers, where due to electrostatic forces, these very thin membranes are made to vibrate. Well, for these applications, we use piezoelectric transducers, even though CMOS are now also being widely explored for neuromodulation, but the fundamental properties of these two are different. So the quality factor of the piezos is normally higher than CMOS, and the output pressure per volt applied is also larger. So a larger quality factor means that the energy will be more concentrated at the resonance frequency, which is good to maximize the focal pressure. And this is the type of signals we use for neuromodulation. They are bursts of continuous waves. So that's why piezoelectric transducers are more suited for this. In imaging, on the other hand, we want to transmit very narrow pulses. So a low Q is normally preferred. So for this reason, I have been focusing my research on piezoelectric transducers, specifically PZT. And the thickness basically much defines the resonance frequency with this equation. And thicknesses are normally in the 100 to 200 micrometer, so 100 to 300 micrometer thick. So then I went to fabricate an array of 26 by 26 piezoelectric transducers on top of my CMOS chip, all them matching the driving electronics. And this is the fabrication flow. So I'm going now a bit more in detail in the fabrication. So we have here pretty much 7 steps. So I start with a PZT sheet. So it's like a wafer size PZT sheet, and I pattern contacts. In this case, it was chrome and gold on top and bottom of these PZT sheets using backside alignment and liftoff. And these contacts will match all of the electrical contacts on the CMOS chip to make a proper connection. So after liftoff, using a dicing saw, I dice this PZT from the backside, just about 20% of the total thickness, and you'll understand why. So after dicing from the backside in two directions to make the bottoms of the pillars, then I do flip chip bonding of the pre-diced or the backside diced PZT on top of the CMOS chip using an anisotropic conductive film. And I'm going to go in detail after. After adhesion, I dice from the top side now. And since I diced from the backside before, now the blade doesn't have to go all the way deep, meaning it will not scratch the CMOS chip. So this way the CMOS chip is intact and not damaged by the blade. So after I have all the pillars defined, I place an epoxy in between the curves of the pillars. And this will provide me with a support then to deposit a top metal, which will be the ground connection. So this is a summary of the process. There's also here a 3D illustration that summarizes what I just said, but just I'm going to step by step to show you some pictures. So the first action is to prepare the CMOS chip. This is the photo of the chip. You see here each square, it's the output of an electronic driver. And you see all these tiny rectangles in between, they are just test pads. And then I have the pads that I want to wire bond to in the end, but I need to cover them with photoresist, otherwise they will be damaged during the process and I cannot wire bond in the end. So the first step is actually protecting these 80 square meter pads with photoresist. So this was a traditional lithography step. The second step is to pattern the contacts on the piezo sheet. So it was a layer of chrome gold. It was by evaporation and liftoff. And you see here the patterns. So I made several in the wafer, and then I just diced individual dies. And the step afterwards was dicing from the backside. When what you see here is already the curves of a PZT sheet diced from the backside using a 50 micrometer thick blade. Then next step was the adhesion. So I have to adhere now this chip to the CMOS chip. And for this, I use an isotropic conductive film, which has 3 micrometer conductive particles with very low density, meaning that they will conduct vertically, but not in, let's say in Z, but not in XY. And this is very nice because I don't have to worry about short circuits in between the pads. As you see here a photo after a test with a glass cover slip. And these pads are about 50 micrometer. And this is done using this addition is done using a flip chip bonder specific to the fine placer lambda. And what you see here is an overlay of 2 images. This is for alignment. So one image is the CMOS chip underneath. You see actually still the white pads, aluminum pads on the CMOS chip. And the other chip is the backside diced PZT. So they are facing each other, but there are cameras pointing at both. So this allows me to align. So you see here that a pad in the CMOS chip looks like this. So the white aluminum output pad and the test pad. And what you see here in the zoom in is actually the white pad and the test pad. And underneath, it's difficult to see, but there is the PZT pad as well, diced from the back. So then after alignment, with a 50 megapascal pressure, 175 degrees of temperature for 15 seconds, these 2 chips are perfectly aligned and bonded. The following step was topside dicing. So after these chips were diced, I went to the dicing saw again. I diced from the top side using the same process as before. And what you see here is the final chip after dicing with already a 2D array of PZT. There's here a microscope photo focused on the top and here on the bottom, where you see that the test pads on the CMOS chip are visible, meaning the PZT was really fully diced. There's here a SEM photo as well. Okay, my mouse is a bit slow. And the final step, or the 2 final steps, were deposit the epoxy in between the curves. So this was done with a pressurized dispenser and a very narrow syringe tip with a Epotec 301-2 epoxy. So it's a biocompatible epoxy that then flows by capillarity through these pillars. And then curing it at 60 degrees overnight, I end up with a cured epoxy. And then this is sputtered a thin layer of the chrome and then alpha micron of copper to make the ground plane on top of the array. And this is the final chip, already wire bonded and encapsulated in a PCB. And then I went on to measure the ultrasound. So I made this test setup with a water tank and the chip inside the water tank. And why water? Water has very similar acoustic properties than the brain and soft tissue. So it's a good model. I positioned a hydrophone that records pressure waves inside of the water, connected to a 3-axis motorized stage. And then I scanned a plane, several planes on top of the chip while recording everything using an oscilloscope controlled by a computer. So first I programmed the chip to output a planar wave. So no focusing, and I obtained this homogeneous pressure map. And then when focusing, this was for 5 millimeter deep focusing at different angles. I got managed to make the focus spot move around just like I wanted. And here's just a 3 dB focus spot, so 3 dB below the maximum to see the dimensions of the focus spot. However, there are still things to improve. And this is now bridging from postdoc to my research group. There are still a lot of things to improve. Focal pressure is still only 100 kilopascal, needs to be higher. Focus spot dimensions are larger than expected, so they are very long still. There are still here things that I don't understand what's going on. Can be the effect of the epoxy between the pillars. The silicon underneath also creates problems. And there's the question, can this already perform neuromodulation? Because of the low pressure, I don't think it can. So just a brief comparison in state of the art. Until recently, it was the only chip that was specifically designed for neuromodulation. There was another one, but it was a PCB level. But very recently, there was another paper 2 months ago published. But still, it's still the one with the largest, highest frequency by far, more than 4 times the frequency, and also the larger pressure per volt at the focus spot. So how can we move forward? So what I have here is for me the ideal transducer array, and I'll try to explain now what's missing from the current version. So you see here the CMOS chip with the transistors and the back end where the connections are made to the piezos. There's first a very big aspect ratio. So in between the pillars, there's normally 30 micrometer spacing or 50 micrometers, depends on the blade, and around the 100 micrometer pitch. And I would like to make this connection from top to bottom. And this is around 260 micrometer thick. So it's a very large aspect ratio to make such a connection, but this connection would allow me to drive the transducer with a bipolar wave, meaning if I can drive with these two signals that are offset in phase by 180 degrees, if I look at the differential between the pillars, I obtain twice the voltage with the same power supply, which means I'll get twice the pressure. However, the aspect ratio makes this connection very difficult, but I have a few ideas on how to improve this. Second, the silicon simulations have shown that it highly attenuates the output pressure. So, but we need the silicon because we need the CMOS chip. So how can we solve this? Maybe thinning the silicon and replacing it by a better material. And also a matching layer. PZT or piezoelectric ceramics are highly mismatched acoustically with tissue, meaning there's a lot of reflections. A matching layer can be fabricated on top to try to mitigate these reflections, but so far it hasn't been done, at least with good levels of transmission coefficient. So these are all fabrication challenges that need to be solved. Just very quickly, this research also led to used to be used in other projects in a nice way. So what you see here is a tiny ultrasonic tag. It works like an RFID tag, but it uses ultrasound, and it's a temperature sensor. So you see a CMOS chip. On top, there's a piezoelectric transducer, and it's around 300 microns by 300 microns by 500 microns. And it can be, so, and the reader for this tag is a conventional ultrasound probe that you can see in the hospitals. And it can power this tag and also read the temperature wirelessly. And what Chen, a PhD student at Columbia, did was then placing two of these ultrasound tags next to a nerve, and then ultrasound was used to stimulate the nerve, and temperature was recorded wirelessly simultaneously. You see here two results, focused ultrasound being applied. These are EMG responses after the ultrasound. So there was EMG results, and then temperature increasing for different intensities of this ultrasound. And this is important for safety reasons to test the effect of ultrasound in the temperature. And this is just another example of flexible ultrasound arrays. So you see here a curved array. This was done by Jeff Eloyan, also a PhD student at the time, that was able to image using also flexible arrays, which is a nice application. So now, very briefly, just go to the applications where I see this can be very impactful. And the first one that I see is peripheral nerve stimulation, specifically the vagus nerve, which is used to treat epilepsy, for instance. So the idea is to make a small chip, which is a patch, first to get the image using conventional ultrasound imaging, detect the vagus nerve, which is always between the jugular vein and the carotid artery, and then focus the ultrasound with high enough precision to focus only on subsections of the nerve that are relevant for the disease. Another application is to do deep brain stimulation, just to place this chip either on the skull of a small animal or just below the skull to overcome the large attenuation of the skull for ultrasound. And then, just like using a controlled remote, change the focal spot anywhere in the brain with good spatial resolution. And this today doesn't exist. The ability to stimulate anywhere in the brain with hundreds of micrometers of spatial resolution where we want, it's a device that doesn't exist yet. Secondly, okay, but if we want to avoid surgery in the brain and still do deep brain stimulation, what you see here is the blood vessels in the brain, arteries and veins, and these specifically have diameters larger than 1.2 millimeters, meaning they can withstand a stent. And placing a stent is a minimally invasive procedure. So my idea is to place ultrasound arrays with CMOS chips as well in a stent, and then have them placed in a specific region of the brain and use beamforming to focus the ultrasound wave, for instance, in the subthalamic nucleus, which is important for Parkinson's. This could be a replacement for deep brain stimulation electrodes without surgery. I'm working with a Phenox, a German company that is providing the stents. Yeah, these are the students currently working on this project. And then for neuroscience, to understand how these things work actually at the neuronal level and also at the ion channel protein level, I have three projects running. So the first one is a brain-on-chip platform. This is with collaboration with Massimo Mastrangeli, a colleague at the Microelectronics department, where he is making 3D electrode arrays on a silicon chip that can record extracellular activity of neurons. And then Gandhi, our PhD student, is fabricating ultrasound transducers that will go on top. And the idea is then to focus ultrasound on neurons and then record the one-to-one relationship between the focal spot, one neuron, and the electrode, to be able to map and sweep the ultrasound parameters and map the network, the single neuron and network responses of, and try to decode what are the optimal parameters for ultrasound neuromodulation. Another project that is now starting is to actually record different types of protein ion channels under the effect of ultrasound. This is a collaboration with Andreas Artel at Columbia and Wojciech Kopek at Max Planck Institute that they are able to either simulate or also actually place protein ion channels on top of chips. And I will design a CMOS chip to read the gating events, so focusing ultrasound on these protein ion channels and then record the gating events and correlate both. And then there's also a project of using ultrasound to promote the transformation of stem cells into neurons, which is normally not very efficient. So we are studying in collaboration with Difna Meyer at TU Delft in Applied Sciences, study the effect of ultrasound in the stem cell to neuron generation. And finally, this is my last slide before the conclusion. I just want to leave this as a food for thought because I still haven't decided what to do with this, but there is the magnetoacoustic stimulation also, where, and this is a recent paper, and there's a sentence there that transcranial magnetoacoustic stimulation adds a static magnetic field based on transcranial ultrasound, so that electrical stimulation is generated by magnetoacoustic coupling while superimposing the effect of ultrasonic stimulation. So this is the idea of using static magnetic fields with ultrasound to be to reduce the amplitudes of required ultrasound to stimulate while maintaining the same depth. So that might be something very interesting to do here by merging magnetics and ultrasound. And so the conclusions, we know that pharmaceutical drugs are not good, and the existing biotonic medicine is still bulky and very invasive. We know that ultrasound is a good prospect, and microchips are the way to get there. And this can lead to several interesting applications that I just described, hopefully interesting for you as well. And that's it. This is my team, and thank you very much for your time.
Thank you, Tiago. Thank you so much for this very interesting presentation, for the nice introduction, and also the state of the art. So I really appreciate it. I hope everybody did. So, and now it's open, the session is open for questions. Does anyone wants to, Professor Paulo, you can start, yes, please.
Sorry, I only have 5 minutes left now, so but thanks Tiago for the presentation. Actually, it was nice to see what you have been doing in Delft. I think we talked maybe 2 years ago or 3 years ago. So I had one question. First is how did you, let's say, define the PZT thickness? It's quite a thick layer. So how did you model it to come to this thickness? And I saw that in the, let's say, the competition work, somewhere there was work done with monolithic integration. And in the work done with monolithic integration, what is the, let's say, the transducer being used? Is it also PZT or, yeah, or something else? It's PZT actually here, it's monolithic.
So actually, this is a mistake. So, okay, I just submitted this paper 2 weeks ago, and I noticed that I put flip chip. I just noticed it now. But anyway, there's a bit of sometimes I have mentioned in papers and proposals about my approach being monolithic, and they always come back to me sometimes saying that no, it's not a piece of silicon, a single piece of silicon, so it's not monolithic, right? But if you look at CMOS chips, the silicon is just the backside. Then they put metals, they put oxides, they put more metals, more oxides. So the definition of monolithic is a bit vague, but let's call my approach flip chip, because indeed, in the end, but in the middle, I'm flipping a chip and bonding. So, but it's also what these other projects do. So they say, but it's the same process. It's also flip chip.
Okay.
Monolithic, to the real sense of the word, is used with CMOS, with MEMS. So those processes are really, even from the foundry, they are CMOS compatible because they use the same materials as CMOS process. So those can actually come from the foundry already pre-made. CMOS. I can go back to the.
But yes, I had the question of the thickness. How did you define
the thickness?
The
so
thickness of the piece
I
of
have
wood?
here the slide where I talked about it. Let me go back. So the thickness is basically this equation for a given thickness. So basically for lambda, thickness has to be lambda over 2. So for a specific resonance frequency that translates to a wavelength, thickness has to be half of the wavelength, basically to allow half of the wave to bounce back and forth in the piezo. And they are quite thick. That's why I buy the sheets pre-made. But there are also sputter targets that can be already bought with PZT, and we can.
I know.
But those are allowed only for very thin layers, meaning for very high frequency ultrasound. If we want to go to maybe 100 megahertz.
And this will limit how deep you can go.
Precisely. So it's a good approach, which I'm going to pursue in the future, to really increase the frequency, to focus ultrasound on cell cultures, for instance, that are just a millimeter or hundreds of microns above the chip. And then we can really get maybe single cell resolution. But then, we have to still optimize this process of sputtering and polling. It's a complicated process. So I still have to work on that in the future.
Okay. I just had a second question, and if I can answer, then I need to leave. At very end, on the integration, you were on the new things you wanted to do. You were saying that you would like to, let's say, to contact. I presume it was through the silicon that we were talking about the large aspect ratio vias. Was this correct? No?
So it can be TSVs, but so when I, for instance, make a hole all the way through. To the silicon, even to the back ends. And so let me just explain quickly. Ideal backing layer is air. So ideally, I have an air pocket between the piezo and the chip. But I also need to make an electrical connection. So I have to have some metal or something that conducts. And these two collide. So I want to try to thin the silicon to see if at least by thinning, it's good enough. Because then maybe it's thin enough, the back end is thin enough that I can still improve transmission efficiency. Otherwise, I might have to maybe explore making some like donut shape electrodes where I have an air gap in the middle, something like this.
And Tiago, using for example, a porous material would help?
Yes, maybe like some kind of bio professor. Thank you for your questions.
Bye. Sorry, we'll talk later.
Yes, of course. So that could help. So I've been reading about kind of foamy materials that can have maybe some conductive particles and lots of air in different densities, and then maybe this can be optimized. Maybe even, and then an interesting thing is that for imaging, if I want to do image as well, then the optimal material is the opposite of. It's not a good reflector, but it has to allow ultrasound to go into the chip and attenuate. So another approach is maybe use either MEMS that allow an air, let's say, in a controlled way, an air cavity to open and close, or even microfluidics, where you pass some material where when you want to have damping, and you have air when you want to have the high coefficient. But this is still very abstract in my head. I have no idea how to do it. I have to think about it.
Okay. Okay. Thank you, Tiago. I think we can extend a little bit more the session for questions if someone has anything else to ask to Tiago.
I have a question for Tiago.
Please.
So first of all, thank you very much for the very interesting talk. My question is, you have very high aspect ratios, and you're going to be applying ultrasounds. Do you run into problems with the mechanical stability of your pillars?
That's a very good question. That was actually also one of the reasons I used the epoxy. So my first reason to use the epoxy was that one, actually. And then I just realized it's also a nice way to have support for the top metal. So it is possible that for very high voltages being applied, the piezo might even delaminate or break. But these materials are able to normally withstand hundreds of volts. And according to the required pressures, 20 to 30 volts might be enough. So it might not be a problem, but yes, that needs to be properly simulated to see if there are like even acute problems or long-term problems. Thank you.
Okay, thank you.
Okay. Does anyone else has any other question?
Yeah, I have one relating to the biocompatibility. So what you said, you wanted your devices to be implantable or in a patch, right? Mm-hmm. So what I don't know, issues do you think you will face to actually allow this integration, because you are using several materials that most probably are toxic for the human body? So how do you plan to do this?
So that is one of the reasons that these type of CMOS are being also explored, because they are biocompatible. And ceramics using PZT, they contain lead, so they are not biocompatible. So one approach is now to start moving more towards these membrane-based transducers, if I want to place it inside the body, or to use, there are some lead-free piezoelectric transducers, such as barium titanate, but they are a bit less efficient. So they have a lower output efficiency. Do I need to drive it with higher voltages to get the same pressure? So I think my first approach would be to do that, because it's a similar fabrication process. I just have to replace the material, and it's commercially available. But depending on the performance, I might also need to explore this type of transducers to be biocompatible. But if you are outside the body, then it's fine.
So I also have one question, Tiago. At slide 17, I guess. So I was concerned about one affirmation that you said, that the mechanism of these ultrasounds, or the effect of this ultrasound in the cells, in the cell membrane, it's not fully clear. It can be cavitation or not. So how can this be confirmed? And I saw that you have a collaboration with this in this group in Delft, that you're trying to promote nerve cells differentiation.
Yes.
So haven't you looked at the effects of this type of mechanism in the cell, or?
That's a very good question. So I think I'm an engineer, and I think the biology needs to answer that question. To really understand the biomolecular mechanisms, I think it needs to come from more of the biology side, but I'm working with biology towards that. But
because this would be very interesting, even in this collaboration that you have, to understand what happens to cell membrane, what happens to cell viability after the application, because it could hamper all the type of applications that you were mentioning. First, we need to look at the side effects of this type of...
True. So the papers that I mentioned that already are looking at the mechanisms, or the groups, not the papers, which are these ones. So this one at Caltech. So all of these studies, they do also, they try to inspect safety. So the ones that do the experiments in vivo, they do histology afterwards, they look at the fibers in the muscle, etc. And they, of course, for very high intensities, they see damage. For intensities that is still leads to a response, they often don't see damage. So there's a safety window, at least that is at least doing histology and other types of analysis. But in these like cellular experiments, so the only one that was actually experimental was this one. Both of these are simulations, and they use basically a camera to inspect to see if they have cavitation, to see if they have pore formation with a very high resolution camera. I did not see anything, and also there is a threshold of intensity of ultrasound that is known to lead to cavitation. It's 720 milliwatts per centimeter square. So if we stay below that level, according to physics, we should not have cavitation. But yes, it's true, we need to be sure. So we need to really understand what's happening, and you are right. So these three projects that I have, that I mentioned, about looking at the neurons, proteins, and differentiation, the idea is also to see what can we learn in terms of the mechanisms, so to make absolutely sure this is safe at the short term and long term.
Okay, thank you, Tiago. Does any other question?
Yes, I have a question.
Okay, please go for it.
It's more like you explained the side. I know some of the side effects for using pharmaceuticals, medicine, but for this kind of technology, I didn't understand if there is any study about the side effects of using this type of devices in the human brain, directly in the human. It is there is a what kind of side effects of long use
that's a very
exposition?
Because maybe this gives the impression that this has no side effects because it's targeted, but that's not true. That's not true. So because unless we have single cell resolution and are only interacting with a neuron that is not working properly in the case of a disease, there's always going to be side effects. Maybe they are not going to be sensed by the patient. But for instance, in implantable vagus nerve stimulators, and this is also true in the brain, there are normally electrodes. This is a cross section of a nerve. There are normally electrodes around the nerve, like a wrapped around, and they stimulate normally many fibers inside the nerve. So for this reason, that you can have in the vagus nerve specifically, it controls heart rate, it controls the intestinal tract, it also has afferent nerves to the brain, and side effects of this can be, for instance, shortness of breath, bradycardia, so low heart rate, or hoarseness, so in the voice. There are still side effects. Of course, because of the implant itself, you can have infection, etc. But if it's non-invasive, as long as we are still stimulating more neurons than we should, there always can be some side effects. But comparing to pharma, we believe it to be less.
It is possible, like the same. When you want only to focus in one three-dimensional point in the body, instead of using one device, using like three devices and triangulate the position of that neuron.
Yes, I mean in a nerve, that's difficult, but the nerves are long, so you could only target two dimensions, not the third. But yes, if you put perpendicular structures, you can at least you can of course improve the spatial resolution in all dimensions. Here you can only do it in the perpendicular planes to the device. So yes, there has been a recent paper that they tried that on the brain, placing multiple arrays to improve spatial resolution, but still it is bounded by wavelength. So the point that you mentioned, it is small, but still not small enough.
Okay, thank you.
Thank
so
you.
I see one raised hand from Felipe Cardoso. Please, can you unmute yourself?
Yes, can you hear me?
Yes, yes.
Very nice presentation, Tiago. I have a question still regarding the effect on the cells, and maybe I panels of the ultrasounds. There are some reports that shows as well that ultrasounds can be used for healing effect of cells, so basically regeneration of tissues. Are you looking at that, or is it already understood the mechanisms of that?
That's a good question, Philippe. Thank you. I haven't checked that. Actually, I know. We both know that your boss, my previous boss, has a big project on that, using ultrasound to heal wounds. I haven't looked into how that works actually, because that's a bit out of the scope in terms of application. But I think the devices that I'm developing can also be used for that, but I have no idea about the mechanisms. Actually, I don't know what's going on in terms of healing. So yeah, that's a good question. I have to look into that. Maybe it's also interesting to explore.
Okay, I think for now we can just say that if there's no more questions, we can just thank very much Tiago for this very nice seminar, and hope that everything goes well in your research, in your research path, with successful achievements for the near future in this area.
Thank you so much, Verónica and Vanya and everyone at INESC MN for inviting me. It was very nice. And yeah, let's keep in touch.